Hello all, welcome to my channel. In this video, we are going to discuss about RNA, types of RNA and their function. RNA, it stands for ribonucleic acid. It is a polymer of nucleotide which is made up of ribose sugar, phosphate group and nitrogenous bases, which is almost equal with DNA, only there is a difference in between thymine and uracil. DNA contain thymine, instead of thymine RNA contain uracil. It is a polymeric molecule essential in various biological roles like coding, decoding, regulation, expression of gene, etc. Most RNA molecules are single stranded, but RNA molecule may contain region which can form complementary base pairing with the RNA strand and loops back on itself. Here is the image shows the loop formation. Here is so double stranded, looks like double stranded helical loop. Here is the single helical loop. So if the RNA will have some double stranded region, the base in RNA are adenine, guanine, uracil and cytosine. Here you see the pairing with G, C, U, A, U, A, again C, G likewise. RNA is a single stranded as you see in the display photograph. The strand has 5' end to 3' end. It is composed of ribonucleotides. The ribonucleotides are linked together by 3' to 5' phosphodiester bond. Next is types of RNA. In both prokaryotes and eukaryotes, there are three main types of RNA, that is rRNA or ribosomal RNA, tRNA or transfer RNA, mRNA or messenger RNA. Messenger RNA, let's discuss first, accounts for about 5% of total DNA in the cell. It is 5% of total DNA of a body. Most heterogeneous of the three types of RNA in terms of both base sequence and size. It is heterogeneous. It carries genetic code copied from DNA during transcription, as it is the messenger RNA. It helps in transcription and it contains triplet of nucleotides called codon. As part of a post-transcriptional processing in eukaryotes, the 5' end of mRNA is capped with a guanosine triphosphate nucleotide which help in mRNA recognition during translation of protein synthesis. Similarly, the 3' end of mRNA has a polytail of multiple adenylate residues which prevent enzymatic degradation of mRNA. Function: mRNA transcribes the genetic code from DNA into a form that can be read and used to make protein. mRNA carries genetic information from nucleus to cytoplasm of cell. Next is ribosomal RNA or rRNA. It found in the ribosome account for 80% of total DNA in the cell. Ribosome consists of two major components, that is small subunit. Here is the two subunit. One Here is one which we know as 70s and 30s. S stands for Sedgwick unit. In this small and larger subunit, large subunit join amino acid to form polypeptide chain. Each subunit comprises one or more ribosomal RNA molecules and variety of ribosomal protein, or shortly known as R protein. Different RNA present in the ribosome include small rRNA and large rRNA, which denote their presence in small and large subunit of ribosome. This complex structure travel along the mRNA molecule during translation and facilitate the assembly of amino acid to form polypeptide chain. They bind to tRNA and other molecules that are crucial for protein synthesis. So, as ribosome are the site of protein synthesis, as the name is ribosomal RNA, it helps in protein synthesis. Next is the transfer RNA or tRNA. It is the smallest among the three other type of RNA, having about 75 to 95 nucleotides. Only tRNA are essential component of a translation, with their main function is transfer of amino acid during protein synthesis. Here the blue portion indicate the attachment of amino acid. As it is attached amino acid in this site, which indicates which protein is going to be formed. So, they are called transfer RNA. Each of 20 amino acid has a specific tRNA that bind with it and transfer to the growing polypeptide chain. tRNA also act as adapters in the translation of genetic sequence of mRNA into protein. As I say, as it add amino acid, one amino acid indicate one protein. So here it translates the genetic sequence of mRNA into protein. Therefore, it also called as adapter molecule. This is the structure. Here is the site, amino acid attachment site, where amino acid added, and here this is the vowel position, anticodon. Here is the anticodon loop and variable loop. So, transfer RNA have clover leaf model structure as you see in the display image. Apart from 4 usual bases, they normally contain some unusual bases, mostly formed by methylation of usual bases. For example, methyl guanine and methyl cytosine. 3 structural loops are formed via hydrogen bonding. OH group, here is the display image. You may see the 3' end. Here is the amino acid binding site have 3' end, and phosphate binding site is the 5' end. 3' end is the attachment site. Center of the loop encompass the anticodon. Here is the anticodon. The anticodon is a 3-base nucleotide sequence that binds to mRNA codon. Suppose here is the C, G, suppose here is the U. So here is the subsequent nucleotides are attached at this site. This interaction between codon and anticodon specifies the next amino acid to be added during protein synthesis, and the function is protein synthesis and transferring to amino acid to ribosome, and that correspond each of the 3 nucleotide codon of RNA. Specifically, it helps in protein synthesis. Here we finish with the RNA and types. If you have any query, please comment in the comment box.